Welcome back, students. Today we will discuss about introduction to cytopathology for medical laboratory technician students. The course code BSc MLT 6004. The course name Medical Lab Technology 2. Myself, Mr. Shankar, Assistant Professor, Department of MLT, Galgotias University. The course outcome: explain patient rights and responsibilities and demonstrate chemical and reagents useful in the sample collection and analysis. Broad understanding of maintenance record inventory test result in cytopathology and histopathology. Learning outcomes: after completing this lecture, the student should be able to describe the difference between the exfoliative and intervention cytology, describe the cytopathology. Introduction: cytopathology is a branch of pathology that studies and diagnoses disease on cellular level. It means students in the pathology, which are the we are going for the diagnostic part in the laboratory under the pathology only. Okay, here pathology has the several branches like histopathology and cytopathology. Okay, in the histopathology, we are going for collection tissue sample from the human body. Okay, whether it is the biopsies, different type of the biopsies available. Okay, in that one, it will take minimum 4 days per reporting. Instead of that one, we are going for the cytology. मतलब we are collecting the cells. Okay, we are collecting the cells. That cells we are going for the observation. How we are going for observation? By the stain. Okay, cytopathology is also called as cytology, which means the study of the cell. Okay, which are the cells collecting from the human body? That cells we are going for observation. Okay, you can see here the cells will be collected by the syringe, then they prepare the smear like this one in the histopathology department. In the cytopathology, the sampling methods, majorly two techniques is there, that is exfoliative cytology, intervention cytology. Students kindly remember exfoliative cytology and intervention cytology. Exfoliative cytology. In this method, cells are collected after they have been either spontaneously seeked by the body, that is a spontaneous exfoliation, and manually scraped or brushed off the surface in the body. It is called mechanical exfoliation. Okay, but under the exfoliative cytology, we have two type of sample techniques is there, that is the spontaneous exfoliation and mechanical exfoliation. Now we are discussing about spontaneous exfoliation. An example of spontaneous exfoliation is when cells of the pleural cavity or peritoneal cavity are seeped into the pleural or peritoneal fluid. This is fluid can be collected via various methods for examination. Now you can see here picture that is explanation of this picture like a pleural fluid collection, effusion pleural fluid collection. Okay, by the some abnormality, by the some abnormality like a liver tuberculosis or liver cancer, during that time the automatically the pleural fluid will be effusion started, accumulation of the pleural fluid. Okay, that fluid we need to collect. Why we need to collect that fluid? Where you are going for a spontaneous exfoliation. Spontaneous means automatically shed out the cells, okay, from the surface area. Okay, in the liver, the liver composed with the pleural layer, pleural cavity. The pleural made up of the two layers, that is internal and external. These both layers will be, it will made up of this number of epithelial cells surface on the surface of this both layers, like epithelial cells is there. That epithelial cells we need to collect, okay, through the fluorosynthesis. Through the fluorosynthesis. After this collecting this fluid, we can go for the diagnosis of this fluid only. Okay. Mechanical exfoliation. Example of mechanical exfoliation include tap smear, where cells are scraped from the cervix with the cervical spatula or bronchial brushings. The students, simple example for the mechanical exfoliation, cells collecting from the female genital area for estimation of the cervical cancer. Okay, now you can see this picture. There, we are applying the brush mechanical pressure on the cells. We are applying the mechanical pressure on the cells to collecting. You can see we are using the cervic brush. The brush we are put it near to the cervical through the vagina by using this some sampling technique like a spatula. Okay, we are going for this best sample you can prepare on the slide for the slide preparation. After slide preparation, we can go for the staining. That time we can detect the cervical cancer, what type of grade, either primary or secondary tertiary grade. Intervention cytology. Intervention cytology is this method. The cells are collected from the lesions or cells masses using the syringes by the application of negative pressure suction in order to increase the yield. Now you can see here in intervention cytology we are collecting here thyroid tumor samples as well as female breast tumor samples by the syringe. Okay, because we are applying the negative pressure on the tumor thick mass lesions, when you get obtain the sample, that's the syringe sample, we can go for the preparation of the slide preparation and staining. Okay, this is about introduction to the cytopathology. Okay, here in this slide, I was added to for the practice questions for you. Apply your knowledge on explorative cytology, discuss on mechanical explorative cytology, and also you can apply your knowledge on intervention cytology. These are the textbooks for you. Textbook of Medical Laboratory Technology by the Godkar and Medical Laboratory Technology by the Mukherjee. Thank you, all my dear students.